Thanks very much for the invitation. It's a pleasure to present our work here in this context. I'm going to be talking about our study looking at neuroimmune mechanism in vivo in the brain in autism with a focus on brain and choroid plexus. And this is work that's conducted in combination between the Martinos Center for Autism and the Martinos Center for Biomedical Imaging at Massachusetts General Hospital and Harvard Medical School. So as we have heard repeatedly between yesterday and today, there's certainly a lot of evidence for the immune system to be implicated in autism spectrum disorder. There's on the one hand genes that are associated with innate immunity or microglia and associative markers that have been implicated. There's also reports of elevated cytokines in some individuals with autism, and there's alterations in some immune cells, for example, natural killer cells, T cell populations, as well as peripheral blood monocytes. And so given these involvements of the immune system, we wanted to see, but what do we see in the brain, in vivo, in autism? And so one approach to be able to do this is using PET imaging, so positron emission tomography, and to study this using imaging. And so the protein that we targeted is called translocator protein, or short TSPO, which is an 18 kilodalton protein that is a mitochondrial protein. So it sits, as you can see there on that image, at the between the outer, in the outer mitochondrial membrane, but close to inner membrane contact sites. And it's a protein that is upregulated during inflammatory processes, which is why it works well, as a marker, as a proxy for inflammation. And it can be imaged with PET, including second-generation radio tracers, which are even more efficient. And this is one of the ones we used. We used PBR28, which binds this target. And so this is what the imaging setup looks like. We use a high-resolution brain PET scanner. And so you can see in the back is the standard 3 Tesla MRI scanner, and then inserted in it, which you can see in the center of the bore, is this PET camera. And so using this simultaneous approach, we get both the structural MRI, gives us the structure, which you can see there on the top, as well as the PET data, which gives us the density and regional localization of the protein of interest. And then given that this scan is acquired at the same time in the same participant, we could fuse these images and immediately have information in terms of structure and protein density, at the same time in the same image. And so for those of you who may not be familiar with, PBR28 PET imaging, I'm showing you here an image, which is a group mean images of a group of controls. And so what we do is we measure radiation that's normalized by injected dose and body weight and normalized to the whole brain mean. And if you were to look at a TSPO PET image in a neurodegenerative disease, such as the one I'm showing you here is amyotrophic lateral sclerosis, or short ALS. And what you can see is this really significant upregulation in the motor cortex. And so this reflects this increased PBR28 binding, reflects an increased density of this TSPO protein in that area. And of course, that's in the area where we would have expected in ALS, given the known motor symptoms of the disease and the postmortem work, which shows that this is the region where you see motor neuron degeneration and microglial activation occurring. So for our study, we included adults because the PET imaging does include, require the administration of a radioactive radio tracer. So it was adults only, ages 18 to 40. We included males and females. They had to have a DSM-5 diagnosis corroborated by ADOS and ADI. And we included individuals across the intellectual ability spectrum. So we started at IQ had to be above 35. And we did exclude for epilepsy, unless it was very well controlled, stable, and no change in medication. The reason why we excluded is because it's known that epilepsy is associated with, upregulation and inflammation processes, and with PBR28, imaging has been shown to lead to, increased signal, so it would have been a confounding factor. The control participants were age, sex, and TSPO polymorphism matched controls. So regarding the TSPO polymorphism match, I have to say that this radioligand is sensitive to a single nucleotide polymorphism in the TSPO gene. And so it leads to participants, when they're scanned with this radioligand, to be low, mixed, or high affinity binders. And so that's something we need to control for. And in addition, we also exclude for low affinity binders, which is a very small percentage, maybe 5% of individuals, because the PET signal would just be too low. It would just be negligible. So we only include mixed and high affinity binders. They had to have normal IQ, and for both groups we excluded if participants were on anti-inflammatory drugs or benzodiazepines, because those would have been confounding factors, and as I mentioned, no low affinity binders. Given that we're including individuals across the spectrum, we make sure, of course, to have a detailed training protocol. They get scanner videos to view at home that go through every single step. If there's a specific step they would like to make sure that they're familiarized with. There's also a procedural video that shows the entire sequence, and we also do on-site training to make sure that the participants are really familiarized with the procedures, the study staff, and very comfortable with the scan before actually proceeding with a real scan where data acquisition will happen. And so what I'm showing you here are the results for a group of males with ASD scanned with PBR28. And so what you can see is this TSPO mean image across the brain, this increase in the thalamus, brainstem, as well as cerebellum, where you see this in red. That is a typical pattern that we see with TSPO. What I'm showing you now is the statistical maps that compares these individuals with ASD with age, sex, and TSPO polymorphism matched controls. And you can see that the individuals with ASD had lower TSPO levels compared to controls in several brain regions that are known to be associated certainly with socio-emotional processing, and also have been previously implicated in autism. And so we saw lower TSPO levels in the temporal gyrus, the angular gyrus, and also both the anterior and the posterior cingulate, and the insula bilaterally. And so I have to point out that there was actually not a single region in which we saw an increase in TSPO in the males. So they only had regionally decreased TSPO compared to the controls. When we scanned females with ASD, this is the pattern that we saw at the group level. And if we compare them with matched controls, we saw elevated TSPO level, specifically in the cingulate cortex and an area of the corpus callosum in the females with ASD compared to the neurotypical control females. And we did this scan both for the males as well as for the female more than once. So we had a repeat after 4.5 months. And at the repeat scan, we saw the same pattern. So we saw this, lower TSPO in the males and higher TSPO in the females. And in the females, there was not a region in which they had lower TSPO. So to summarize, we saw really this diametrically opposed pattern in males and females, but in each case, they significantly differed from the controls, with lower levels in the males and higher levels in the females with ASD. Now, of course, what does this mean? The TSPO biology and the roles with which it's associated are pretty complex, and there's certainly several. It's a mitochondrial protein that plays a key role in different processes that are important for mitochondrial function, bioenergetics, metabolism, potentially steroidogenesis. And as such, with in vivo imaging, while the strength is we now know which protein is altered and where in the brain, we actually don't know, in which cell types this would be in the human in vivo. And also the process may, of course, be different, in the neurodegeneration context compared to in a psychiatric disorder such as ASD. So all we know is that we have definitely identified a biological process which, something is different between ASD and controls, but it could very well be both disease-specific and sex-specific. But clearly it implicates, this mitochondrial protein in ASD biology. Now, that was very focused on the brain, and now I want to make sure I step also into this area where a lot of the immune function may actually be occurring in specific regions that are at the brain border. And so this is certainly a rapidly growing field, but which really highlights that the meninges the bone marrow of the skull, as well as, for example, the choroid plexus play these key roles because they are these niches, these compartments that are within the brain, but really at the border, where there is more interaction between the immune system. And so I want to zoom in on the choroid plexus, which is known as the immune gatekeeper of the brain. You may know it as the CSF production site, so the cerebrospinal production site, because that's certainly one of its key roles. It is really there at the interface between systemic circulation, so the blood, and the central nervous system. And you can see in this area here that it's a highly vascularized structure. So the choroid plexus of the lateral ventricle would be here. And it really acts as an immunological barrier that has dynamic permeability. And so we wanted to look at the choroid plexus in individuals with autism. For imaging, that means that we need a clearly delineated structure that will be our region of interest. And for the choroid plexus, the automated pipelines are currently not optimized enough. And so they are now better, and we now have some that use deep learning. But when we conducted the study, we chose to do manual tracing, which was at least still considered the gold standard. And so I'm showing you here what this looks like if we render it in 3D, the region of interest that we had for this choroid plexus that we decided to look at in ASD and matched controls. And so what we see is if we're now looking at these images and are focusing specifically on the choroid plexus, you can see there, whoops, on the top right, that in the males, we don't see a change, we don't see a difference between the controls and the individuals with ASD in terms of TSPO levels in the choroid plexus. In the females, on the other hand, we really see significantly higher TSPO levels in the choroid plexus in the females with ASD compared to the matched controls. And in some females, this was actually really very significantly different from what we ever saw in controls. We then also looked at the volume of the structure. And so what I'm showing you on the top right there are the males, where we saw a significant enlargement of the choroid plexus volume in the males with ASD compared to the controls. In the females, we did not see these cross-structural changes. That being said, the mean was still numerically larger, and we have a smaller group of females, so we can't exclude that with a sample size we could be underpowered to detect this, but we definitely see a change in volume in the males. In the males, where we do see this change in volume, we looked at disease severity that we had assessed based on the social affect and restrictive repetitive behavior score from the ADOS, and correlated that with the volume. So in this case, it says CP volume over ICV. We have done the investigation both with and without the normalization to this intracranial volume, ICV, and it does not change anything in our case. So the normalization factor just allows us to control for head size in case that were to affect choroid plexus size. And what we see is a correlation between symptom severity and the size of the choroid plexus. So individuals who have a larger choroid plexus had more severe symptoms. So this shows that we have both volumetric as well as mitochondrial alteration in the choroid plexus in ASD. And of course, the volume change that we see, may be a reflection of a cumulative event, because all we see is this, in adult, because we're imaging in adult. And the TSP alterations that we see in female could reflect immune metabolic or mitochondrial alteration. We know that they're certainly mitochondrial because it's a mitochondrial protein that we're imaging. And so these changes, both in terms of enlargement and TSP elevations, have also been seen in other neurological and psychiatric disorders. I have to say it's a very active area of research right now, so most of these studies are fairly recent. But in MS, there is an enlargement of the choroid plexus, so in multiple sclerosis, and also in depression. Recent reports show that there is enlargement of the choroid plexus in patients with depression compared to controls. And so if we zoom out a little, this pathway blood from the choroid plexus to the CSF and then going from the CSF to the brain is really being rediscussed as potentially having been underestimated and needing to be studied in more detail to see if this could be a route of propagation in the context of inflammation. In the context of preclinical work, what has been done in autism relevant models is a lab from Dr. Lettinen at Boston Children's. They looked at the choroid plexus in the maternal immune activation model, so the model where the dam is exposed to poly-IC during pregnancy, which leads to autism-relevant symptoms in the offspring. When they studied the embryo, they saw that there was an increased number of macrophages that were present in the CP. And so this certainly illustrates that in the context of autism-relevant symptoms, earlier in life, there can be alterations that are relevant for the immune system that do happen in the choroid plexus, and suggest that the choroid plexus certainly could be a key area to study in autism. So to summarize, our work shares, I think, novel insights into the involvement of the choroid plexus in autism, based both on volume and mitochondrial alteration. And we saw sex-specific effects in terms of mitochondrial protein levels, both in the brain and in the choroid plexus. And so I think this highlights the need to continue to better understand the choroid plexus and underlying biology in autism. And with that, I'd like to make sure I thank my mentors, Dr. Jacob Hooker, who is the scientific director of the Lurie Center. Dr. Christopher McDougall is the director of the Lurie Center. Jane Seng is an instructor in my lab who used to be a postdoc in my lab and led a lot of the TSPO work in females. And I also want to make sure I acknowledge Lind Lila, who together with Jane led the choroid plexus work. And of course, special thanks to all the participants and their families, because without them we couldn't do this research. And the funding for this specific study came from the Robert and Donna Lundgren Fund that funds our neuroimmune studies in autism. And thank you for your attention, and happy to take any questions.